Okay. Hello, Brian.
Hey, Eli.
Tell us your sort of place in the world of parasitology.
Sure. So, um, by trade and profession now, I'm a high school teacher, and I'm currently occupying a role in teacher leadership. So my actual title is new teacher support specialist. So,
Mhm.
since my days of parasitology, I've kind of veered in a different direction. But it's really thanks to parasitology that I wound up in this direction at all. So, um, how did I get there? I did undergraduate research in parasitology at Eastern Illinois University, and I studied worms that live inside of quail. And then I stuck around and got a master's degree in science, and I studied parasitology for my master's as well. During that time, I loved my parasite research, but I really loved teaching more. And so, parasites were the vehicle by which I got into teaching through my graduate studies.
I see. And during your research, what kind of sort of hands-on project did you get to do?
Excuse me. Yeah, so I did two kind of like major research projects with parasites during my studies. In my undergraduate years at Eastern, like I said, I studied what are crop worms inside of the crop of quail. Basically, I was looking at pen raised bobwhite quail and asking the question, what types of parasites exist within these kind of like agricultural settings for for quail, and then how does it affect their numbers, and how does it affect their care and management, and so on and so forth. And so I was looking at prevalence and abundance studies, and at first it was just a survey trying to figure out what types of worms might exist or what types of other parasites, intracellular or maybe things circulating in the blood. And what we really found was that there was a particular parasite called capillaria that lived inside of the crop of quail, and in these agricultural settings it could spread very quickly, and it could wind up infecting a whole lot of them at a really high abundance within each individual, to the point where there would be so many worms inside the crops of these things that they would starve to death because they couldn't get any food down their digestive system,
Wow.
because their crop was just overflowing with worms. Like, as soon as you do a necropsy, you could start seeing them like coming out of the neck, and it was pretty brutal. But yeah, there would be hundreds, if not more, in a single individual. And it was pretty gnarly, pretty disgusting, and it's fecal oral transmissions. So since quail in that setting were just like on the ground, they were just pecking in all of each other's poop, and they were just passing parasite from individual to individual, and it was problematic. So that was a really cool study, and that was study one. That was undergrad, and that went well. I presented at like a regional conference and stuff, and my advisor asked if I wanted to stick around and keep on studying parasites. And so I was thinking about going to Northern Arizona University for graduate work in forestry, but I got this offer and had already started in on parasites and thought it would be fun, so I stuck around. And then my second research project was looking at what's called the giant American deer liver fluke, which is Fascioloides magna. And I was looking at so this liver fluke is a really weird one.
Okay.
Like many flukes, it has a really complicated life cycle where, um, so if you start at the adult phase, it hangs out with like at least one other adult inside of a necrotic cyst inside of a deer, inside of a white-tailed deer, and it will mate with itself, and it will mate with the other, and it will make bunches and bunches and bunches of eggs that get deposited out through the fecal matter. And then those eggs will hatch inside of water, and they'll become a miracidium. And the miracidium is like a tiny little larval stage of the parasite. It'll swim through the water until it finds a snail, and then it will enter the snail. And inside of the snail, it will asexually reproduce, effectively castrating the snail to the point that there are lots and lots, and then they bust out of the snail and they go and insist on some reed or something like that in like salt marshes, some plant matter that a deer will eventually come by, eat, and then the cycle begins again. So the larval stage burrows through, gets to the liver, makes a cyst. And so I was looking at the part specifically where the miracidium enters the snail. And so I did microscopy, and I did sectioning, and we did some behavioral studies and stuff like that.
Sure. That's
Yeah.
So then, does that mean that the deer is like the intermediate host for the snail, or is it the other way around?
So it means that the deer is the primary host for Fasciculoides magna, and the snail is the intermediate host.
Oh, okay.
And in this, yeah, and so in this system, there's one intermediate host. In other parasites that are like this one, there may be two intermediate hosts.
Okay.
But the defining feature is in intermediate hosts, they will undergo asexual reproduction. And in the primary hosts, they're able to reach sexual maturity and undergo sexual reproduction. Now, these ones are hermaphroditic, so they'll self and other.
Aren't these the flukes that, like with most blood flukes or just flukes in general, don't they have like one of them is kind of like a canoe shape and the other one like fits into the groove and then they just like mate for life? Or am I just thinking of regular blood flukes?
I believe you're thinking of schistosoma moniasis. And so, with schistosomes, they do that. But that's kind of a special romantic feature of schistosomes. Yeah.
Okay.
Yeah.
So that's cool. So then when you were working with these snails, like what did this look like in like a lab setting? Like what were you doing?
Yeah, sure. So it was a pretty cool workflow and pretty ingenious on my advisor's part, I think, because what he managed to do was figure out a research system wherein he could utilize the eggs from the adult phase to be the fuel for all of the research, which is huge because these things are so fecund. They make so many eggs that if you can get like a single, a deer liver that's infected, you could have enough research fuel for like a year, no problem. So what we would do is in November in Minnesota, they do population control hunts for white-tailed deer, and so we would go up there, and basically we would be at the checkout station. So anytime somebody bagged a deer, we would ask them. Well, we'd actually catch them on the way in, and we'd give them a bag, and we'd say, "Hey, when you got your deer, can you bring the liver to us?" And then when they would check out, they would bring us the liver, and then we had like a dissection station there, and we'd slice through the liver. We'd find the necrotic sacs that housed the deer liver flukes, and then those deer liver flukes, they give off this like what we call black gold, and their black gold is just filled with eggs, millions of eggs probably. So we'd collect all of this, like black sputum essentially from these livers, and then we'd separate them out and we'd grab all the eggs and we'd bring them back to our lab. And then so those eggs could just sit in the refrigerator in like a dormant state until you hatched them. And so there was a hatching protocol. But that's what I mean, it was so cool, right? Because you could go get 1 million eggs, bring them back, have them sit in the fridge until you're ready for them. Sure. And so what it looked like in the lab was that we would you'd start in the morning, you'd hatch a bunch of eggs, and then you'd do whatever kind of protocol you were up to. So like one of my protocols was watching as the eggs hatched and the miracidium emerged. I had a microscopy experiment where I'd watch them just in the dissecting scope, and after some trial and error with engineering, like you need to get the snail in such an orientation where you can watch as the miracidium burrows into the snail. And so you kind of need to like expose their soft parts or keep them still or something. So I tried a bunch of different ways, but ultimately just wound up using a double-sided piece of tape and just sticking them on their back to the bottom of a petri dish essentially. And then just put some miracidia in, and then we would watch. And so I had a matrix of like behaviors. Did they bump into it? Did they stick for a few seconds? If they stuck, did they start to like burrow in? And the point of that was like looking at a few different species of snail that were either same genus, different species, or a couple out of genus types, and just seeing like, was there difference in behavior? Did they recognize that one type was infectable versus another one not so much? And how does that kind of affect like that very early interaction between parasite and host, where parasite is trying to initiate that burrowing type of infection? So that was one thing we did. Another thing we did was we were looking at so we figured out that Miracidio can't infect a different genus of snail, particularly a different genus that lives in the same area. So particularly limnaeid snails are infected by these Miracidia, and Physa snails live in the same ponds. So we were trying to see like, why don't they infect Physa? Did they try? Or what's the deal? And what we found was that they do actually try, but Physa mucus is incredibly toxic to Fascioloides magna Miracidia. And so like to the point where we would this all sounds kind of funny in retrospect, right? But we would like take the snail and I'd massage their little head foot until I could get mucus off of it, you know? And so I'd massage enough head foot to get enough mucus that I could kind of like dilute into the toxicity series, and then expose the Miracidia and kind of see what happens. And to a pretty low dilution that I don't remember anymore, but that Physa was extraordinarily toxic. Like it blew up all of them, microscopically speaking, where on one slide you would still see them swimming. On another slide with the Physa mucus, they'd be like a bunch of little blobs, like it blew up, you know, like, so it was a pretty cool finding, you know. The Physa mucus was a really effective barrier to infection. And so, you know, between the behavioral side and that side, and then the histological side that I can tell you a little bit more about if you'd like, we are just kind of like looking at the same issue from a few different perspectives and seeing how they relate.
Sure. Yeah. So it was like sort of a test of the different kinds of snails and how they react to this parasite. That's super interesting.
Yeah. Yeah, like, yeah, well, it's an interesting question, right? Why can't this parasite just infect other species? It would certainly be an evolutionary advantage if you could get into more hosts.
Yeah.
Because that's the problem when you're a parasite, right? It's like, how do I find my host in order to continue my life cycle? And in a lot of cases, that's really complicated. I mean, these Miracidia are, you know, tinier than your fingernail, and they're supposed to go find specific species of snails living in ephemeral salt marshes and stuff. Like it's amazing that they're able to. How do they do it? You know,
that's the coolest thing about studying parasites is they all have this, like, to be a parasite is to answer the question, how do I make a relationship with another organism where I get something from them at their expense? And that's just, they each have their own unique way of answering that question.
absolutely. Yeah. And some of them are like, well, I'll make as little impact as possible. And some are like, I'll kill you.
Yeah.
You know, and yeah,
Yeah,
exactly.
sure. Like a tick on the back of a rhinoceros is so different than like, you know, a Toxoplasma gondii that has, you know, 2 phases and could even control humans maybe, you know, stuff like that is super, super impressive. Do you have a favorite parasite?
Oh, it's an interesting question. You know, I think that familiarity probably stoked my bias towards my favorite being the one I knew most about, you know.
Okay.
So I really liked Fascioloides magna. It's such a cool parasite. I mean, yeah, the phase I was looking at was this tiny microscopic phase, but when it's in the deer liver, it's one of the largest parasites of all. It's huge. I mean,
Really?
this big, some of them maybe this big individuals, right? And it was just, I don't know. I think what's so cool about biology, like in general, is that the closer you look, the more you find.
Mm.
And I would always tell my students that, like we'd get a little bit of soil or something, be like, is this alive? Well, actually, it contains thousands of organisms. How do we find them? How do we get them to show their face? And so I think that what I love so much about fascioloides was just that I dug deep enough that I've just seen some stuff that probably like a handful of people on the planet have seen, and uh, I love it because of the familiarity. Now, in terms of like weirdness, there are some other ones that you've already mentioned, like
Okay.
cordyceps and stuff that are way cool.
Yeah.
So it's hard to choose because I love the multi-host cycle. So like
Mhm.
any trematodes I think are really cool. Generally, I like the trematodes a little bit better than the roundworms, just because their life cycles tend to be a little bit stranger and more mystery.
You're a helmet kind of guy.
Yeah.
Okay, sure, sure. Yeah, I was I think Toxoplasma gondii is like the entry point for a lot of people, but then the Gordian worm or horsehair worm,
Oh, sure.
super interesting. The Saculina parasite barnacle with the crabs, that one I was learning a lot about.
Oh, tell me about that one. I don't remember.
It's basically this barnacle that its host is a harvester crab, and so basically what it does is it uses the sharp points of its barnacle shell and kind of almost turns into a syringe basically, and so it'll sort of pierce their underbelly and then inject its soft gelatinous body in, and then
Oh, that's awesome.
based on if it's a male or female harvester crab, it does one of two things. If it's the female harvester crab, it can, I think, basically just hijack the eggs that are already in there for itself. And then if it's a male one, it suppresses its endocrine system and makes the crab think it starts acting like it's pregnant almost, and it starts just collecting all this food for the parasite so that it can kind of asexually, make its own continuum that way. So that was super cool for me to hear about.
Wonderful. Wonderful. Yeah, the diversity of strategies that parasites have figured out to infect organisms is just amazing. And
Mhm.
and I think that people don't really recognize how common parasitism is. I mean, there's you've probably read about it, but there's a claim out there, and I lend it credence, that essentially says every single organism on the planet has at least one specific parasite, meaning a parasite that cannot infect other organisms or other species. At least one. And so, if that is true, then the outcome is that parasitism is the most common life strategy on the planet, and they are the most common organisms on the planet, which is pretty interesting. And I think is likely true. Yeah.
Sure. Yeah. I mean, there's all these little like sayings sort of like that, and it kind of makes you think the like entire world's buildup of what sort of animals are actually out there. Assuming there is one parasite or at minimum per person, and then there's that other one about how 1 in 4 organisms is like always a bat or something like that. You start to think like what really is building up our entire Earth's ecosystem.
And I think that it's revolutionary once you start to realize that everything is based on symbiosis, whether it's parasitism, mutualism, or commensalism, or whatever. But
Mhm.
I mean, like in a human being, for every one of your cells, there's 100 fungal bacterial cells.
Mm.
We are symbionts, just like every other organism on the planet is. Some of them are small enough to be the symbiont itself, of course, but symbiosis is everywhere, and parasitism is just a really cool version of it that you get lots of gnarly stories to tell your friends about.
Totally, and that's this whole project for me. I mean, I got to interview earlier somebody who was talking about maggot debridement therapy and just all these cool also applications of parasites to the medical field, and that's been its own journey too. So
Yeah,
I'm,
sure.
I'm really seeing the whole breadth.
Have you Have you stumbled upon the hygiene hypothesis in
Oh
your
yeah,
research?
yeah. I found that pretty interesting. I was also hearing about, these people who are trying to get hookworm, or, walking around to get these, allergen suppressant parasite effects. And, I was kind of advised to kind of steer clear of that. Yeah, I mean, it's fun to research in theory, but then like when it comes down to the science, sometimes it's not all there. But that was what was super interesting to me.
If you haven't checked it out, so yeah, sure, there are people who get hookworms from, band-aids and stuff
Mm.
that farmers in Mexico send up, or even Santa Cruz. I think I used to show a video about that. But there's also a fair amount of legitimate research that goes into it too. There are people at University of Iowa, I believe, maybe it was Iowa State, but I think it was University of Iowa. They were looking at how whipworm could be utilized, intentional infections utilized to treat both MS and Crohn's disease, I believe. But they had really promising results, albeit this was maybe 8 years ago. So I'm sure that they have updated studies and stuff since then. But we saw them talk at conference, and holy cow, it was amazing.
Of course.
You'd infect people with whipworms, which is a direct life cycle, so they're easy to treat. If something got out of control.
Hmm.
But just the infection with whipworm resulted, at least in small trial experiments, with the decrease of lesions of in the brains of people with MS. And their whole idea was that immunoglobulin G, this whole set of immunoglobulin of antibodies and stuff, is set up to fight parasitism, but they don't have enough parasites in developed countries. And so as a result, they end up fighting random allergens and
Yeah.
antigens that just come on in from the environment, or even your own body cells. And so I thought that was so cool. I wonder if they have new stuff. They were trying to like isolate specific chemicals that may be responsible for occupying the immunoglobulins. So I'm not sure if they wound up doing that, but it was promising.
Yeah, it's exciting. I can't wait to hear, what even the next 10 to 5 years look like in terms of, furthering our sort of new look into parasites in medicine. I mean, because the earlier sort of ideas of parasites in medicine, they don't date back too far officially, but then when we look back at how we even discovered things like maggot debridement therapy or even medical leech therapy, it comes back to like certain world wars when they, saw people in the trenches with wounds, and the people who had maggots inside of them, seemed to be surviving and the others weren't. I mean, it's so cool to find the history of things, but still we're
Certainly.
really at the brink, and there's so much more ahead of us, and I'm really excited to see what that looks like.
Yeah. Well, and with those two, that's a genre, but I think like what the hygiene hypothesis gets at is this
Yeah.
idea that parasites and their hosts have co-evolved for so long that when you pull one from the other, you wind up with unintended consequences that just are surprising. Like you think parasite, I don't want that, a tapeworm, that sounds terrible. But it may be that having low level parasitic infection actually keeps you healthier overall by maintaining the various systems that have evolved to maintain reasonable parasitic populations. Tapeworms don't really hurt you all that much.
Of course.
They might cause a vitamin B deficiency or
Yeah,
something,
maybe.
but they're mostly just trying to hitch a ride without causing too many problems, so they can just bask in your like nutritious juices at a nice warm temperature.
Totally.
Um, so so yeah, it's just interesting, like we feel like we've come a long way in eliminating parasitism, but what were the unintended consequences of doing so? Is it diabetes and other autoimmune problems? Maybe.
Who knows? Yeah. I, that's so interesting to me too.
If if you overlay like the incidence of, if you overlay maps that show the incidence of autoimmune disorders and the incidence of parasitism in human populations, which generally broadly correlates with like developing versus developed countries, you find that developing countries don't have these autoimmune disorders. Is that because of parasitism or the lack thereof? Maybe.
Yeah, yeah, I was hearing about that too. Like even some languages don't even have certain words for like allergies or things like that, and it's just almost sort of pointing towards this bigger theory of that. Yeah, super interesting stuff.
Yeah, that's interesting. Very interesting. Yeah. Yeah.
Well, it was great talking to you. Thank you very much.
Yeah, you too.
And
Absolutely.
I hope you watch. I'll send you a recording or maybe even the link to my presentation if you want to see it.